Jai Sri Ram, Jai Jai Sri Ram. Hello everyone, how are you all? My name is GYANENDRA, I am biology educator in India. In this video, we will discuss mechanism of vision. Yes, we are going to see mechanism of vision. Okay. For understanding mechanism of vision, we must know the structure of eye that I have already discussed into another video, and I will share the link into description. From there you can see that video also. Fine. In this video we will discuss mechanism of vision, and before going the mechanism of vision, we will discuss structure of the retina also. Retina, we all know retina is the innermost layer of the eye, and this retina is the layer, we are calling it neural layer. This retina is the only layer where some specialized cells are present. That cells are called cone cell, and then that rod cells. This cone cell and rod cells, is the light sensitive cells. They actually photosensitive cells. They absorb light. Yeah, they absorb light. Fine. And then one knob is coming out from the eye through a region. That region is called blind spot. Yes, that region is called actually blind spot. And this blind spot is the region of the retina, yeah, this blind spot is the region of retina where rod and cone cells are absent, right? And when rod and cone cells are absent there, so that region cannot absorb light. This is the blind spot region, because here rod and cone cells are absent. That's why we cannot visualize from this region. Fine. Now in the structure of the retina, we observe that in the structure of retina, one groove like structure present. Yes students, we observe one groove like structure present. This groove like structure is called, in the structure of a retina, we observe that there is one groove like structure. That groove like structure is called fovea centralis. Fovea centralis. Fine. Now this fovea centralis is surrounded by, like suppose this circle is fovea centralis. This fovea centralis, is the surrounded by one region. That region is called macula lutea. Macula lutea. Fine. This macula lutea, is very rich in cone cells. Cone cells. Okay. We will see structure of the cone cell. we will see structure. We will see about cone cell in detail. And this fovea centralis is the region. See, it is rich in cone cell. It doesn't mean that it have not rod cell. It have rod cells also, and it have cone cells also. Fine. But in the center where this fovea centralis present, this fovea centralis is the region, where only cone cells are present. Only cone cells are present. And this fovea centralis is the region, which is for sharpest vision. If light will strike there, the resolution of the light, the resolution of the image, the resolution of the object will be very, very, very clear. Fine. Then you see, there is some region of this retina where this rod and cone cells are not present. In this region, specialized type of cells are present. The specialized cells are called melanocytes. This melanocytes, actually they have one pigment that is called melanin pigment. This melanin pigment is black color pigment. Fine. And it will actually, it will prevent scattering of the light in our eye. Fine. When you see the second layer, you see the choroid. You see choroid is actually full of the melanin pigment. Very, very, very black color layer, hyperpigmented layer. Fine. So the scientists believe that this portion of the retina actually, which is rich in this melanin pigment, is perhaps modified choroid region only. Now the point of transition where one side rod and cone cells are present and other side melanocytes are present, this region is called ora serrata. Ora serrata. Ora serrata is the transition point of the retina and choroid. Okay, fine or not? Okay, so next to ora serrata, anterior to ora serrata, we have melanin pigment. Fine. Now we will see different type of cells present in our different type of cells present in our retina of the eye. Fine. So if you see different type of cells present there. Yeah, one point I forget, that this in blind spot region, our optic nerve means cranial nerve second, which is coming out from the eye. They will take all kind of stimulus from the eye, sensory stimulus from eye, and they will take it to the visual cortex of the brain. Fine. Now see, in the retina we will see, in this retina, if we will go from inside to outside, we have first layer of cell, that is called ganglionic cell. Okay, then we have another layer of cell, that is called bipolar cells. First ganglionic cell layer means from outside, from the inside to outside, then bipolar cell, and then next to that we have a very important layer of cells which have photosensitive cells, light sensitive cells, and that is rod and cone cell layer. So outermost in the retina is actually rod and cone cell layer, and after that we have choroid layer. Fine. After that we have choroid layer actually. Okay, let's see that same structure in detail here. So this is the choroid region, this is the choroid layer, and then this is the pigmented epithelium of the retina, okay, which have melanin pigment also. And then we have rod cells, right? We have rod cells, and then next to that, now I'm taking it from outside to inside, and then next to that we have bipolar cell layer, second layer, and then third ganglionic cell layer. Okay, ganglionic cell is innermost layer. They will absorb, this rod and cone cell will absorb light. After that they will generate impulse, that impulse will come to this bipolar neuron will also generate impulse, and then that impulse will call to ganglionic, it will go to ganglionic cell will accept that stimulus, and then they will take it to the visual cortex of our brain. And visual cortex of the brain, according to according to their memory, according to prior information, they can detect the type of object. Fine. But what about this rod and cone cells? This rod and cone cells, a great difference is there between this rod and cone cell. This rod and cone cell, this rod cell have, this rod cell have purple red color pigment, and that pigment is called rhodopsin pigment. And this guy have, cone cell have violet color pigment, that is called iodopsin. Let's compare and see what is this rhodopsin and what is this iodopsin. Fine. This rod cell have which pigment, students? Rhodopsin pigment. Okay. Cone cell have iodopsin pigment, right? This rod cells are nearby 100 million in each cell. Cone cells are only 5 to 6 million in every cell. Okay, they are 5 to 6 million in every cell. 5 to 6 million in every cell. Then this rod cell is for dim light vision. Dim light means low light vision. Okay, and this is for bright light vision. This cone cell is for bright light vision. Okay, like you see in daytime, when light is enough, okay, you can observe the color of the object. Like my pen, my pen is blue color, my notebook is green color, and outside trees and all, they are green color. Easily you can detect the color of the object. Okay, so the color of the object you can detect only by this cone cells. They are for color vision. Fine or not? But night, when light and all everything is off, can you see the color of the object? No, you cannot see the color object. You will feel when light is not there, electricity is not there in the house, you will feel just, oh my, okay, fine, some object is there and it's blue, like it's not blue, sorry, it's like a white red color, something white, black and white color. So this rod cell is for, is for black and white vision. Yes, they are for black and white vision. Is it clear to you all? Rod cell is for black and white vision, cone cell is for color vision. Now what is present inside this rhodopsin? Actually rhodopsin have two parts. First part is called retinal, another part is called opsin. Okay, in corn cell have also iodopsin, they have also retinal, and they have also opsin. Fine, they have also opsin. This retinol, is the originated from the vitamin A. Yes, they originated from, they synthesized from vitamin A, and we all know vitamin A synthesized from carotenoid. So retinol is same in both iodopsin and rhodopsin. Fine or not? But difference is present between opsins. Here opsin is different. Here opsin is different. Okay. In corn cells, opsins are three types. One, which absorb, which causes red color vision. Okay. Another, which causes green color vision. Another, which causes blue color vision. That means here iodopsin have three types. One which is for visualization of red color, another for green color, and third one for blue color. That which one is for visualization of red color, we are calling it erythropsin. Erythropsin. Fine. This one is chloropsin. Chloropsin. And this one is synopsin. Synopsin. High wavelength vision, obviously red color have more wavelength. Middle wavelength vision, okay, and this is for low wavelength vision. Okay, and by this three color only, more than 70,000 of combination we can make. Fine. Where these all pigments are present in rod and cone cell? In the cytoplasm of rod and cone cells, you see some membranous structures are present. In this membranous structure only, this rod and cone cells are integrated. Fine. This rod and cone cells, where they are integrated, students, they are present in this membranous structure. See here. Okay. In this rod and cone cell, this rhodopsin and iodopsin present in this all pigmented structure. Okay, this pigmented structure actually is present in this all membranous structure. It is like it is like thylakoid membrane. If you remember, in the chloroplast, thylakoid membrane was present, and within this thylakoid membrane, chlorophyll was integrated. In same way, in their cytoplasm, so many disc-shaped structures are present. They are membranous structures, and within this membranous structure, rhodopsin and iodopsin is integrated. Hope my point will now clear to you. So this is what difference between rhodopsin and iodopsin, and this is what difference between rod and cone cell. Rod cell is for dim light vision, for black and white vision, and our cone cell is for bright light vision. Fine. Okay, fine. Now we will see mechanism of vision, how we can absorb the light. So actually the light will come like this. Okay, and then light will, the story will start from this rod and cone cell. Rod and cone cell, they absorb light. Okay, they are the cells who absorb light, and after that they generated impulses. That impulses will come to bipolar neurons, and from bipolar neuron it will come to ganglionic cells. From ganglionic cell it will go to the it will go to the visual cortex of the brain. Okay, let's see how. Let's see, we have to discuss it into detail. Actually, if I am taking our rod cells, in the rod cell, our rhodopsin pigment present. Okay, in this rhodopsin pigment, retinol present. This retinol actually is present here in the cis configuration, 11 cis retinol configuration present. Fine. And then we have one protein, opsin protein. This opsin protein is attached to this retinol. Yes, 11 cis form. When light will absorb here, when they will absorb the light, actually this 11 cis retinol convert into all trans retinol. Oh my god, all trans retinol. And this all trans retinol immediately it's separate from opsin. Now this opsin is no more attached to the retinol because configuration is changed, structure is changed. It's converted into all trans retinol. Okay, so opsin is separated here. Before that opsin, it was attached to retinol. Okay, and that will actually generate potential here in this rod cells. Okay, that will generate potential, and that potential, that potential will generate action potential in our bipolar neuron, and from bipolar neuron, not action potential exactly, that is something we are calling the EPSP, we are calling EPSP. Actually it's actually receptor potential, or we are also calling it graded potential. So receptor potential and graded potential will generate. Now what is action potential, what is receptor potential, what is graded potential, what is excitatory postsynaptic potential, what is inhibitory postsynaptic potential, that and all we will see in another video. But yes, potential was generated, graded potential was generated, and then it will generate action potential into the ganglionic cells, and from ganglionic cell the whole information will go to our visual cortex of the brain. Fine, we will see that point in more detail. Like, see, this is our, suppose this is our rod cell. Okay, this is our rod cell. This is our rod cell. In this rod cell, this is the disc. In this disc actually rhodopsin protein present. Which protein present? Rhodopsin. Rhodopsin present. And then in the membrane of these all, rod cells, they have different type of pump, different type of channel. Now there are some channels, they are called sodium ion channel, calcium ion channel. This sodium ion channel and calcium ion channel, they will allow entry of sodium ion and calcium ion. Fine, fine. In general, this is how it's happened. The sodium ion and calcium ion will continuously, it will enter inside the cell, and it will maintain internal charge plus 40 millivolt. Yes. Now for the action of this pump, one secondary messenger is required, that is called cyclic guanosine monophosphate. This cyclic guanosine monophosphate, it's synthesized from GTP. And who will synthesize it? Who will synthesize it? GTP will convert into cyclic GMP, and who will synthesize it? It will synthesize by guanyl cyclase. Guanyl cyclase is one enzyme present in the membrane which will convert GTP into the cyclic GMP. Cyclic GMP will come and they will bind with these all pumps. Yeah, cyclic GMP will come and they will bind with this all pump, and this pump will allow sodium ion and calcium ion, and this rod cells, ROD cells, sorry, rhodopsin pigment. Okay, so this rod cells, it will be positively charged from inside. Now when light strike, here, then the whole scenario only change. How? Light striked here, you see, light striked here. Okay, now this rhodopsin immediately is converted into all transform, and now this retinol, all trans retinol, all trans retinol and opsin separated. Now this opsin, they will activate one protein. That protein is called transducin protein. Fine, what they will do, they will activate one protein that is called transducin protein, and this transducin protein activate one enzyme that is called phosphodiesterase enzyme. Phosphodiesterase enzyme. And this phosphodiesterase enzyme actually, it will break cyclic GMP. Yeah, they will break cyclic GMP. Now what will be result of that, you tell? Light striked rhodopsin separated into all trans retinol and opsin separated, opsin activated transducin and transducin activated phosphodiesterase. Phosphodiesterase immediately they broken all cyclic GMP. Fine. And when they broken this all cyclic GMP, then this channels through which sodium and calcium ion was entering, it stopped working. And when it stopped working, this rod cells or even cone cell is also present. This rod cells actually, what they did, they release a neurotransmitter here. Yeah, they release neurotransmitter here. Which neurotransmitter they release? They release glutamate. Okay. When light was not there, the secretion of glutamate was less. Okay, but when light striked, students, when light striked and the sodium ion channel and potassium ion channel, everything was stopped, then this glutamate secretion, it was decreased. Okay, when light was not there, glutamate secretion was more. Glutamate is neurotransmitter, its secretion was more, but when light is not there, its secretion decreases. Okay, now when the secretion of glutamate will decrease, it will actually generate graded potential into the bipolar neuron, and this bipolar neuron secrete lot of glutamate. Now in the presence of light, it will secrete lot of glutamate. Who will secrete lot of glutamate? Our bipolar neuron secrete lot of glutamate, and then this glutamate will take that, they take that graded potential in our ganglionic cells. Okay, this ganglionic cells, they are cells of our cranial nerve second. So the stimulus will come here, and then they will generate action potential. They generate action potential, and this action potential, it will go to visual cortex of the brain, and visual cortex of the brain, it will decode the all information. Fine, I am repeating everything again, try to understand it. Light striked, okay, here rhodopsin converted into retinol, all trans retinol and opsin. Okay, now this opsin activated transducin, this opsin activated transducin, this transducin activated enzyme phosphodiesterase, and this phosphodiesterase, they broke in all cyclic GMP. When they broken all cyclic GMP, entry of the sodium ion and calcium ion was stopped. When entry of the sodium ion and calcium ion was stopped, then now less amount of neurotransmitter was secreted from this bipolar neurons, this rod cells and cone cells, and when they secrete less amount of glutamate neurotransmitter, then they generated graded potential or receptor potential in the bipolar neuron. Okay, and when they generated receptor potential in this bipolar neuron, they secrete lot of glutamate here. They are forming synapse with our ganglionic cells. Fine, and then this ganglionic cell generated action potential, and then this action potential, it went to our brain, and the things was visualized according to previous information. In our brain we have previous, we have already some information. On the basis of that information, on the basis of that memory, the things was visualized. Now there is a term that is called color blindness. What is color blindness, students? This color blindness is actually X-linked recessive disorder. X-linked recessive disorder. Fine, X-linked recessive disorder, and it is due to mutation in the gene present on the X chromosome actually. So obviously, it will affect more man. And here in this color blindness, mainly there will be problem in the visualization of red and green color. Fine. Then we have one disease that is called nectalopia. Nectalopia means night blindness. Oh my god, night blindness, night, visualization will not be very clear. Okay, and reason of this is what, less vitamin A. Less vitamin A. Okay, when A is less, obviously retinol secretion synthesis will be also less, and when retinol synthesis is less, rhodopsin is less, and when rhodopsin is less, obviously it will cause night blindness. Night blindness have one more case, uh, that have, okay fine, that have different region. Here rod cell will degenerate. Okay, rod cell, there is one disease, is called retinitis pigmentosa. In this retinitis pigmentosa, rod cells will degenerate. Rod cells, they will degenerate. When rod cells will degenerate, obviously it will cause night blindness. Night blindness. Fine, clear or not? Now one student asked me that, sir, what is difference between visualization of the object between the rod and cone cell? See, in the rod cell, because it is for dim light vision, acuity will be less, visual acuity, it will be less. Okay, what about edge detection of the object? What is about edge detection of the object? Obviously edge detection of object will be also less. Color you can see by rod cell, obviously we cannot see the color of the object. Fine. So this is what our mechanism of vision. In this video we covered structure of the retina, and then we covered that how ectern potential generate, and then we completed mechanism of vision. Fine, remaining we will see in next video. Bye.